Hello everybody, I am Dr. M. Sanjeev Kumar. Today we'll study about malaria in detail, its etiology, pathogenesis, clinical features and the treatment. Malaria is one of the major public health problems of our country. Around 15 lakhs of confirmed cases are reported annually by the National Vector-Borne Disease Control Program. Malaria is caused by a microorganism belonging to the genus Plasmodium. There are 5 species of Plasmodium which causes malaria, but 4 species are very important which cause infections in human. These species are Plasmodium falciparum, Plasmodium malaria, Plasmodium ovale and Plasmodium vivax. Vast majority of deaths are caused by Plasmodium falciparum. The remaining 3 species caused a milder degree of malaria in humans. Now we'll study about the pathogenesis of malaria. For any infectious disease, there will be 2 hosts, a primary host and a secondary host. Primary host is also known as the definitive host. A primary host is the host in which sexual reproduction takes place. In case of malaria, the primary host is a female Anopheles mosquito. It acts as a vector of Plasmodium. The secondary host in case of malaria is the man. The primary host being the female Anopheles mosquito, it carries the sporozoid, a motile infectious form of the Plasmodium. When the mosquito bites a human being, it injects these sporozoids into the blood of the human being. These sporozoids, they travel from the blood and enter into the liver in the first. They first undergo a multiplication phase in the liver in the hepatocytes known as tissue schizogony. In the liver, after the tissue schizogony, the merozoites are produced. These merozoites, they enter into the bloodstream and then they attack the freshly formed red blood cells. In the red blood cells, they undergo another level of multiplication. And then the merozoites which are produced, they enter into 2 phases. The first phase of merozoites, they enter back into the liver again and they infect the hepatocytes and undergo a tissue schizogony. The other group of merozoites which were formed in the RBC, they enter into the blood and they transform themselves into micro and macro gametes which are picked up by the female Anopheles mosquito. And then these gametes undergo fertilization in the female Anopheles mosquito and then the sexual reproduction takes place. And the sporozoites are formed which are the infective stage to the humans. Now we'll discuss about the clinical features of the malaria, that is the signs and the symptomatology of the malaria. Fever is the most cardinal feature of the malaria. The fever is often accompanied by headache, chills and rigors. The fever can be intermittent with or without periodicity and it can be continuous too. The other features accompanying malaria are lymphadenopathy, ear discharge, skin rashes and diarrhea. Sometimes the malaria can be non-specific too. It might mimic some viral infections or an enteric fever too. Now we'll study about the complications of malaria. We all know that malarial parasite affects the RBCs. When the RBCs are affected, they get lysed and this leads to the formation of anemia. Sometimes the malarial parasite can enter into the brain and affect the brain. This might lead to coma, seizures and in very rare cases, death occurs. This is known as cerebral form of malaria. The other complications of malaria include liver failure, jaundice, kidney failure, hemorrhage, splenic rupture and sometimes dehydration too can occur. All the clinically suspected cases of malaria should be immediately diagnosed using microscopy and/or rapid detection tests. Microscopy is the gold standard for the confirmation of malaria. Microscopy includes one thick and one thin blood smear. The thick blood smear tells us about the presence of malarial parasite. The thin blood smear confirms the species of the malarial parasite. The rapid detection tests are based on the presence of circulating parasitic antigens. The rapid diagnostic test kits are expensive and temperature sensitive. So these tests are done only when the microscopy details are not obtainable within 24 hours of sample collection. Now coming to the treatment of malaria. In all the clinically suspected cases of malaria, we need to perform a microscopy test. If the microscopy test results are positive, then we decide the treatment according to the species. If it is Plasmodium vivax, we start the treatment with chloroquine for 3 days and primaquine for 14 days. If it is Plasmodium falciparum, we start with ACT, that is artesunate sulfadoxine pyrimethamine, and add primaquine as a single dose. If the microscopy details are not available within 24 hours of sample collection, then we do rapid detection tests for detecting the antigen of the parasite in the blood. If these tests are positive, then we start the treatment with ACT, that is artesunate, chloroquine and pyrimethamine with primaquine as a single dose.